Continuation with the nuclear fusion of the embryo sac. This is the second type of nuclear fusion which takes place in the embryo sac. The second meiotic division, 3 out of the 4 nuclei formed fused to form a triploid nucleus at the chalazal end of the cynomagaspore, where the fourth nucleus remain free at the micropylar end. These are of two types. They are Fristrillaria type and Plumbagillae type. In Fristrillaria type, both the nuclei, that is one haploid and one triploid, undergo two division, giving rise to 4 nuclei at each pole. The 8 nucleated embryo sac consists of a 3-celled haploid egg apparatus, 3-celled triploid antipodals, and a central cell with 2 polar nuclei, 1 haploid and 1 triploid, that is 3 plus 3 plus 2 arrangement. Second type is the Plumbagilla type. In this type of embryo sac, the 2 nuclei, 1 haploid and 1 triploid, divide 1 to form 4 nuclei, that is 2 haploid and 2 triploid nuclei. These 4 nuclei are arranged in a 2 group of 2 nuclei each. One group is present at the micropylar end, consisting of 1 haploid egg and one haploid upper polar nucleus. The other group present at the chalazal end consists of 1 triploid antipodal cell and 1 triploid lower polar nucleus. Hence, the polar nuclei comprises 1 haploid and 1 triploid cell. This type of embryo sac was first observed by Fengerlin in 1938 in Plumbagilla micrantha, special type of tetrasporic embryo sac. The special type of embryo sac was discovered by Martinoli in 1939 in Chrysanthemum cinerifolium. The cynomagaspore contained 4 nuclei arranged in 1 plus 2 plus 2 1 arrangement, that is 1 at the chalazal end, 1 at the micropylar end, and the 2 in the center of the cell. Further development may occur by 2 methods. 2 central nuclei do not fuse but remain closely associated. The 2 nuclei at the micropylar and the chalazal end undergoes 2 division, forming 4 nuclei at each end. The embryo sac now consists of 10 nuclei. The 4 nuclei at the chalazal end form the antipodal, while out of the 4 nuclei at the micropylar end, 3 form the egg apparatus and the 4th nucleus associated with the central cell to form the polar nuclei, that is the 4 plus 3 arrangement. 2 central nuclei fuse to form a diploid nucleus. The cynomagaspore now contains 3 nuclei, each of which undergoes 2 mitotic division, forming 12 nuclei arranged in a 3 group of 4. At the chalazal end, 4 nuclei along with the 3 nuclei from the central cell form the antipodal, whereas at the micropylar end, 3 nuclei form the egg apparatus and the 4th nucleus associated with the diploid nucleus of the middle group to form the central cell. The embryo sac consists of 7 antipodals, 3 egg apparatus and 2 central cell, that is the 7 plus 3 plus 2 arrangement. The additional 3 diploid nucleus present in the antipodal region are called additional antipodals. Hostorial nature of the embryo sac. In majority of the plant, the embryo sac derives its nourishment from the surrounding nuclear tissue. However, in member of Santellaceae and Lawrence, hostorial behavior of the embryo sac is of common occurrence. The synergid and the antipodal give rise to give out hostoria, or the micropylar chalazal end may grow out into their surrounding sporophytic tissue and function as a hostoria. The extension are referred to as micropylar or antipodal ceca. In the extreme case, the entire embryo sac may grow beyond their ovular tissue and get lodged directly on the placenta. Example in Centellum. In member of Lawrence, the embryo sac grow upward into the style up to a various height. The largest embryo sac has been observed in Moculinella rubra, where the embryo sac tip grow up to the style, touches the stigma, and then curve back and grow downward into the style. In such case, fertilization may occur either in the basal region or at certain distance above in the styler region. The haustoria development from antipodal and rigid has will be described further.